Hi everyone, it's Dr. Launderville. This is online podcast number 6, last one before the test. I'm going to talk to you about molecular immunology, which is a whole course. We're just scratching the surface here. Unfortunately, we lost Dr. Holda last year, who offered an excellent immunology course. I suggest that you take one in your future studies. But I do want to spend some time on it because it brings together a lot of this pathways that we've talked about so far, and also it's obviously germane to our current situation with the pandemic, and super fascinating, but complicated. So please take it slow, take some breaks while you're listening to this podcast, and feel free to ask questions during office hours or by email. Okay, so you have many aspects to your immune system. Most people think about the immune system in terms of antibodies and cells and that sort of thing, but there are other aspects to your immune system, one of which is just your outer integument, your skin. So preventing stuff from getting in. These are mechanical defenses. Mucus is also a mechanical defense to bind up pathogens and get them out of your body. The surface area of everything that is exposed to the environment in a human is about 4,000 square feet, which is pretty impressive. That would be, like a good-sized home is 2,000 square feet. But that includes the lining of your gut and nasal passages and so on. If we're talking about just the skin, it's 20 square feet. And that is not a trivial defense, immune defense. So germane to our current situation is you're getting all this advice about always wash your hands. It's because you have a mechanical defense of your skin on your hands, that you may have virus on your skin, but that doesn't mean the virus is going to attack you unless you give it a way into your body, like rubbing your eyes or touching your face, your nose, so on, which would be an entrance to a membrane that the virus could be imported into. So washing your hands. Also, if you have any kind of cut on your hand or open sore on your hand, that's a breakdown in the mechanical defense and another way for pathogens to get in. So in this situation, in our pandemic situation, if you're going someplace like a grocery store where and you have a cut on your hand, you should be wearing gloves on your hands to protect yourself. Okay. If the pathogen makes it past the mechanical defense, like your skin, there are also chemical defenses, like the low pH of your stomach, like enzymes in tears that can inactivate pathogens. If it makes it past those defenses, then there are protein systems that are collectively called innate immunity. This is the system that you're born with, that are cell-based and also cell product-based, so protein-based, and we'll talk about that. And then there's acquired immunity or adaptive immunity. These are where antibodies come in, and these are synthesized in response to an infection. Acquired or innate immunity starts becoming active within minutes to hours of an internal infection, and whereas adaptive immunity takes longer than that. We're going to talk a lot about white blood cells, or leukocytes is another name for that. They're very diverse. in the kinds of cells that they are. So from stem cells, you can get myeloid progenitors that lead to erythrocytes, red blood cells, mast cells, myeloblasts, megakaryocytes, basophils, neutrophils, eosinophils, monocytes, and then macrophages. These are cells that can move in and out of circulation and search for pathogens and ingest them, kill them. From a common lymphoid progenitor, you can get natural killer cells. We'll talk about those. Small lymphocytes become T and B cells, and then B cells, once activated, become plasma cells. These are part of your adaptive immune system. This is part of your innate immune system. So not all organisms have an adaptive immune response. Insects only have the innate response. Plants have only the innate response. No antibodies made in plants, no antibodies made in insects. Okay, here's an H5P question for you. Okay, in your circulatory system, your heart generates blood pressure, and that blood pressure forces non-cellular fluid out of the blood. That fluid's called lymph, and it bathes cells and collects antigens as it does so. Antigen is just anything that you can have an immune response to is considered an antigen, and that includes things that are well known and also things that have never existed on Earth before, like SARS-CoV-2. The antigens collect in lymph nodes. You know that that's these little green nodules here. You know that there are some underneath your jaw. You know that there's some in your armpits. These are collecting points for lymph, where cells of both your innate and acquired immune system will gather and be exposed to these antigens. When you are infected, there can be a lot of cells in those lymph nodes, causing them to swell. And one of the things that a physician will examine if someone's complaining about a possible infection is if their lymph nodes are swollen. They'll feel under your jaw, maybe in your armpit. Also, another thing where you've probably heard about lymph nodes is if you hear that someone has been diagnosed with cancer, and then they will usually try to type the cancer and find out at what stage the cancer is, and find out if the cancer has gone mobile or is metastatic, has moved from its original site. One of the assays they do to determine that is to look at lymph nodes, because lymph nodes will also collect cancer cells once they are metastatic. So if you hear about someone and you hear that their lymph nodes are clear, that's a good sign. That means cancer was caught relatively early. First, we're going to talk about the innate immune system, and then we'll talk about the acquired immune system. In the acquired immune system, we'll talk about B cells and T cells. B cells are named for bone marrow, where they develop, and T cells named for the thymus, which is this little gland high up in your body cavity where T cells mature. Okay, so if we zoom in on a lymph node, you have dendritic cells, which are part of your innate immune system. They can collect antigen throughout the body in different tissues. There also may be soluble antigen that is not being presented on a cell, but it's just in solution in lymph that will come into the lymph nodes. There are B cells and T cells in the lymph nodes. These B cells and T cells come from bone marrow or the thymus and delivered by the circulation to these lymph nodes. These B cells and T cells can be exposed to antigen in lymph and get activated. They can be exposed to soluble antigen in the case of B cells, or presented with antigen from another cell, like a dendritic cell here in the case of T cells, and then increase in their maturation. Later on, we'll talk about B cells making antibodies and T cells helping B cells, so-called T helper cells. But the B and T cells are part of the acquired immunity, and we're going to stick with the innate immunity for now. So these dendritic cells that are patrolling your tissues and picking up antigens and bringing them to the lymph nodes for interaction with the acquired immune system. Another part of your innate immune system is the complement system. The complement system is a cascading series of proteases, a protease cascade. We saw that a protease cascade when we talked about apoptosis. So one complement is a protein, becomes activated, and then that activated protease activates another protease, and then another protease, and so on. That is the classical pathway. This works very similar to like blood clotting pathway. There are also antigens that may have mannose binding lectins on their outside of their surface, like bacterium. These might activate proteins called MASPs, which will interact with the complement system, leading to the production of C3 complement. And then there's an alternate pathway here that also leads to production of C3. We're not going to talk about that alternate pathway. This C3 complement, once it gets made, leads to the production of these complement proteins, C5, 6, 7, and 8, 9, which form a membrane attack complex, essentially pokes a big hole in the surface of whatever the antigen could be. So if it's a bacterium, pokes a big hole in the bacterium. If it's a enveloped virus, like SARS-CoV-2 is an envelope virus, pokes a big hole in that envelope, and that lets good stuff from the pathogen's point of view out, which can kill the cell, and also lets bad stuff in. We're going to talk about some organism killing things like granzymes that can get in through this membrane attack complex. So that's a direct way. By activating the complement system, you can kill the pathogen. An indirect way is that the production of these complements, the cleavage fragments of C3 and C5, are potent chemoattractants. So a theme that you'll see in molecular immunology is that cells will release chemoattractants. Often those are proteins, and those proteins are released locally, and neutrophils, white blood cells that are in the area, will be attracted to those signals and accumulate in that area. And then they release more chemoattractants, and more things get attracted to that area. So it's like calling in the cavalry. Say, hey, there's a problem here. Let's call in other parts of the immune system that deal with this problem. Okay. Eventually, this leads to inflammation. So you know inflammation as redness. Let's say you have an infected cut or something like that. That cut would be red. It would be swollen. It would feel warm to the touch, and there would be pain. So the swelling is caused by vasodilation. More fluid is leaking out of blood vessels and bathing those cells, causing swelling. More cells are attracted to the site because of the release of the complement system and chemoattractants. Because there is more blood supply to that site, that increases the heat carried by the blood in that site, and pain is caused by specific proteins or specific molecules like leukotrienes that are released at that site. You can ask Dr. Shriver about that. She knows a lot about that. Okay. It doesn't have to be because of infection. Also can be because of mechanical injury, like you get a bruise or something like that. But this diagram is talking about a bacterial infection. So this bacterium here has been ingested by a dendritic cell. That dendritic cell is going to present those antigens to T cells down here. There's also some bacteria that have degraded and created soluble antigens. Again, antigen is anything that can elicit an immune response. Neutrophils have been called to the area. Natural killer cells have been called to the area. Maybe some antibodies have been called to this area. In the lymph node, that dendritic cell is going to present antigens from that bacterium to T cells. Those T cells are going to activate B cells. A B cell that is activated has a huge increase in endoplasmic reticulum activity. That's what this little chevron pattern here is, increased in ER activity, and starts pumping out antibodies at a tremendous rate. Okay? Later on, we'll talk about what antibodies do for you. Well, I can talk about it right now. So an antibody has a specialized area at the tips of these Ys. They really are Y-shaped molecules, and at the tips of these Ys is where they bind their antigens. And where they bind those antigens can physically block some part of the pathway. So, for example, in SARS-CoV-2, the coronavirus, the coronavirus is named because of those spike proteins on the outside. It could be that the antibody binds to the spike and prevents the spike from binding to an ACE receptor, and therefore mechanically stops it from entering a cell. If so, that would be called a neutralizing antibody, prevents the reaction that the pathogen needs, prevents that from happening. That's called a neutralizing antibody. When you're testing for antibodies, it's really hard to know which ones are neutralizing and which ones aren't. You just get a total antibody number, and we'll talk more about that at the end of this podcast. Okay? So that's one way an antibody can be good in this situation. Another way is it can tag a cell saying, come kill this one. So if a macrophage is going by and it sees an antibody attached to a cell, it'll ingest that cell, it'll kill that cell. Again, this part here, the dendritic cell and the complement system, part of the innate immune system. T cells and antibodies and B cells, those are part of the acquired immune system. Okay, more about antibodies, which are part of the acquired immune system. They have this Y structure. There are light chains and heavy chains. There are different chemical preparations of antibodies that when you were looking for antibodies, let's say for your lab proposal for self-phys, you may have seen some of these things like FAB fragment or FAB prime 2 bivalent, and they're just saying if we took this antibody prep, and we treated it with papain, which is a protease. It'll cut right there, and then these are the fragments that you get. And if we cut with pepsin, these are the fragments that you get. And depends on the application whether or not this would be okay. Where it's going to bind as antigen is going to be on the ends of these Y's here. There are different oligomerizations of antibodies, and some of these are more prevalent in some species than others. IgM is a huge oligomer of antibodies. There's IgA, which is a dimer of antibodies, IgE, IgG. Most of the time when we're talking about antibodies, like antibodies we use for a Western blot, we talk about IgG, this form of antibodies. But when you make antibodies, custom make antibodies, say in a rabbit or in a mouse, any of these are possibilities. And in fact, a lot of times you don't even know what you're getting unless you specifically try to determine what form it is in. And there are applications where it's really important to know what form. Like, for example, there are applications where only the IgM form will bind to your target. A more realistic picture of an antibody, but you can see the general Y shape, light chain, heavy chain. The CDR, complement determining region, this is essentially the binding site for the antigen. Okay? And one of the amazing things about antibodies is that a person can have over 1 million different antibodies. And by different antibodies, I mean different sequences of that protein. If you were going to sequence every antibody that person makes, even up to 2 million, you would get up to 2 million different sequences that would definitely be an antibody, recognizable as an antibody, but not the same antibody. So how is that person in one genome? So a person only has a single genome, if we're not talking about the mitochondrial genome. And that genome has about 25,000 genes. How is it that you get up to 2 million different antibodies with only that many genes? And it's very cool how it works, actually. So if you look at the amino acid sequence of the light chain, okay, there are, if you start at amino acid 1, go through amino acid 110, for example, in the height amino acid chain, there are amino acids that are pretty much the same no matter what kind of antibody you have. But then there are these hypervariable regions, HV, in that they will be very, very different from one copy of the antibody to another copy of an antibody. And there are several of these, and these are in the binding site, complement determining regions. Okay, so how do you get all those different kinds? Well, it's a lot like CRISPR, actually. So, in the genes that make up the coding region for antibodies, there are these different domains of the gene called V, J, and C. C for constant. And in the maturation of the cells that are going to make antibodies, these are B cells, during the developmental process in which these B cells mature, there is a rearrangement of the V and the J regions. In the light chain, it's one random V combines with one random J to make a functional VJ region. Okay? And they show on this diagram, just for simplicity, 3 V's and 2 J's, but it's up to 40 different V's that could be chosen at random to be combined with just a few J's, different kinds of J's, to make a functional VJ region. And also what's important in this maturation thing is that this arrangement is not close enough to the enhancer part of the gene for this gene to be transcribed, but after they recombine, it is close enough after the DNA has been rearranged. Okay, same thing happens on the heavy chain. A random V, a random D, and a random J come together to make a VDJ region that is functional, and that it will make a protein. Well, potentially make a protein, and I'll show you what I mean for a second. Okay, so how does that happen? Okay, here's the V. It's chosen at random. And then here's J. This is if we're talking about the light chain, or DJ if we're talking about the heavy chain. Okay. Step 1 just shows you the arrangement of the genes. Step 2, there is a protein called RAG recombinase that makes a single-stranded break on each of the strands in this region in between, okay, the V and the J segment, okay. That leaves a free 3' hydroxyl on each of the strands, and that free 3' hydroxyl combines with the opposite strand to make a loop, okay. So this guy combines here to make this loop, this guy combines there to make that loop, and then the intervening sequence that gets cut goes away, just degrades. Then what happens is there is either a symmetrical cut made, and it's made on the J segment right in the middle, so equal top strand and bottom strand. On the V segment, it's made asymmetrical, so that the bottom strand is going to end up being longer than the top strand. So that leaves a overhang on the V region. On the J region, there is a terminal deoxynucleotidal transferase that's going to add random nucleotides to the end. Okay? Then DNA polymerase makes the complementary sequence for the other strand, and then a ligase puts them back together. So hopefully you'll remember how CRISPR works, and this part here is very similar, actually, to how CRISPR works. And what's really interesting and important to this is that the added nucleotides here are random, and so literally it could synthesize anything. Any of the possible codons could go in there randomly. But what it makes is not necessarily going to be in frame. So back here when I said it's going to make a functional VJ region, only 1 out of 3 times will the coding sequence be in frame. Because the codon is 3 nucleotides long. If it makes it on the number, so the number 1 codon is in frame, then you're all set. But if it makes it according to the number 2 codon or the number 3 codon, it'll be out of frame and it won't produce an antibody. The rest of the sequence will also be out of frame. So 1 out of 3 times it'll make an antibody. 2 out of 3 times it won't. Okay. And after all this, then this coding sequence is going to be in reach of the enhancer, this thing here, and that allows the gene to be functional. Okay. So this is in the maturation of B cells, the antibody-producing cells. This recombination of the DNA happens. Okay. Here are the RAG recombinases that are doing the cutting. Okay. They're only made in white blood cells. And if you don't have a functional RAG recombinase, you are severely immunocompromised. So that you essentially don't have any acquired immunity because you can't do this rearrangement of your antibody genes, so you can't make a big series, literally millions of these random antibodies. Not only that, where you have back here, where you have the added nucleotides being random, once you get it all put together, there's activation-induced deaminase. So when the B cells get activated, which we're going to talk about later on in this podcast, by T cells, some of the nucleotides in the sequence are going to change. So the sequence that you put together, some of those will change by this activation-induced deaminase. Okay, and that happens. The change happens every time that B cell divides. Those mutations either make the affinity of the B cell for the antigen, the antibody that the B cell is making for the antigen, either makes it stronger or weaker. But only the ones that bind to the antigen and make it stronger are the ones that get selected and stimulated to proliferate by clonal selection, and we'll talk about that in a second. So there's all these different antibodies made by recombination, and then after you select a B cell, you still play with the sequence a little bit more to see which among these sequences is going to bind to the antigen best, and only the B cells that are making antibodies that bind to the antigen best are the ones that are stimulated to make lots more antibodies. Okay, another H5P question for you. Okay, so what do I mean by clonal selection? We talked about this when we talked about monoclonal and polyclonal antibodies. Okay, so maturation of the B cell, you make all kinds of variety, millions of different kinds of B cells that are making antibodies. Okay, these are actually antibody receptors. Antigen-specific receptors, not antibodies, but B cells are also making antibodies. The ones that are selected by T cell activation, we'll talk about that soon, those are the ones that get stimulated to turn into plasma cells and make lots and lots of antibodies. They will be stimulated to divide and then turn into plasma cells and make lots and lots of antibodies. So the antigen is sort of shopping among all these different possible antigen-specific receptors, finds the one that binds to best, then the T cell activates that, and away you go. Okay? All right. As I mentioned before, when plasma cells get activated by T cells, it's sort of in the way of this arrow there, they become plasma cells and they'll pump out lots and lots of antibodies. And only the ones that have high affinity between the antigen and the antibody receptor does this happen. And so the antibodies that come out should be high affinity antibodies. They don't bind in a wimpy way, they bind in a very tight way. And a plasma cell is no slouch at making protein. So a mature plasma cell has been stimulated by a T cell, can secrete thousands of antibodies molecules per second. Okay? And needs lots of ER to process all that protein. Thousands of antibody molecules per second. Okay. So, as we mentioned before, antibodies part of your acquired immune system. How are they going to help? They could neutralize the antigen. They could literally block a binding site so the pathogen can't go through its life cycle. It could tag a cell for recognition by another kind of leukocyte, of which are macrophages and also these natural killer cells, I'll talk about in a second. Those are part of the innate immune system, not part of acquired immunity, but they can interact with elements produced by the acquired immunity. Antibodies are produced by the acquired immunity system. Okay? So, if you get an antibody tagged on you, that kills those cells. There's all kinds of chemotherapies now that are monoclonal antibodies. Antibodies that have been designed and selected for binding particular epitopes, antigens on cancers. The person is given those antibodies. Those antibodies seek out those particular cancers, bind to those cells, and then your innate immune system, which includes macrophages and natural killer cells, recognize that and kill the cells. Now, natural killer cells don't need antibodies to recognize a stress cell and kill it. Leukocytes do. Well, that's not completely true, but leukocytes can use antibodies to recognize cells. Natural killer cells are inhibited from binding to normal cells. They can recognize stressed or cancer cells or virus-infected cells and activate killing systems. So perforin, which is a large pore-forming protein complex, so you can open up a hole in the cell like we saw with the complement system, or granzymes that once they get into the cell, they act like proteases and kill the cell that way, similar to apoptosis. They can also secrete interferon gamma, which is important in fighting against virus and the viral life cycle. Interferon gamma is one of the things that has been tried as a therapy for COVID-19. And we talked about interferon gamma being activated early in the early phase of COVID-19 disease. Okay, so now we've moved on to the innate or the acquired immune system, talking about B cells and T cells. And we talked about B cells getting help from T cells, but how does that happen? Okay, there is a thing called the major histocompatibility complex. It is on T cells and in other cells, and it interacts with B cells. Once again, B cells mature in bone marrow, T cells mature in the thymus. Both are part of the acquired immune system. Both are considered part of your white blood cells or leukocytes. So, in any cell in your body, that has a nucleus, proteins are constantly being broken down, and then pieces of those broken down proteins are presented on the outside of the cell, bound to this thing called the major histocompatibility complex. And it got its name from, it was discovered when they were trying to figure out why some tissue grafts were accepted by the recipient organism and some were rejected. So histocompatibility has to do with tissue graft. That's just the origin of the name. Okay. Again, B cells, T cells, in B cells in bone, T cells in the thymus, and spleen also is important in maturation of the lymphocytes. Okay. So, in most organisms, talk about major histocompatibility complex. In humans, the major histocompatibility complex is called HLA instead. It's just a different name. It means the same thing. Okay? So, for example, if you know of anybody who is interested in being on a donor list for, say, being a kidney donor or something like that, they will have their HLA complement typed to find out if they're a good match for a potential recipient. And HLA just means major histocompatibility complex. So HLA proteins, aka major histocompatibility complex, are just like antibodies. They have a different shape, but they have a lot in common with antibodies in that there's millions of them. They are produced by recombining DNA in this very same way that we saw with antibodies to create those millions of different types. One individual is going to have a different complement of HLA than another individual. It doesn't mean they don't have anything in common, but altogether they're going to look different than another individual. Same thing with antibodies. But HLA, aka MHC, are membrane bound. Antibodies are not. Okay. For human population, there's 2,000 different alleles in the human population. So there's a lot of variability out there. So normally we think about alleles as there's one version you got one from mom, there's one version you got from dad, but there may be lots of versions in a population. So either you got one eye color or another eye color from your mom or your dad, but among humans there's lots of different eye colors. Among humans there are 2,000 different alleles for HLA and molecules. Okay, and that means that two humans are very unlikely to match, and that's why organ donation is tricky, and that matching somebody for an organ donation is a complicated process. All right, two different classes of MHC proteins. I'm just going to say MHC from now on, even though HLA is the human version of MHC. There's class 1 and class 2. Class 1 is taking antigens from the cell itself and putting it on the surface of the cell. So it's going to bind an antigen, a piece of a protein, say, not necessarily a protein, but let's just say protein. It's going to put it right up there. It's going to go all the way through the Golgi, go up to the cell surface, and then it's going to present that right on the outside of the cell. Say, hey, look at this antigen, okay? And they are going to interact with cytotoxic T cells. And those cytotoxic T cells will either recognize that piece of protein as something that came from the cell that is normal. This is a normal part of the cell, in which case they won't do anything. Or they will interact with that thing and say, hey, this does not look like a normal part of the cell. And then they will kill it. They're called cytotoxic T cells because they kill it. Okay? Then there's class 2 MHC proteins. And they take things that only came from the outside of the cell. Okay? So if this antigen originated from outside the cell, it gets presented on a class 2 MHC protein, one of these guys over here, and only professional antigen presenting cells have class 2, and these interact with T helper cells. Okay, T helper cells are going to interact with B cells, they're going to make antibodies. Okay? So, let's say if you are a virus and you got into a cell, and that cell took apart that virus, and that virus showed up on the outside of the cell in class 1 MHC, kills it because it interacts with cytotoxic T cell. Let's say that virus came from the outside of the cell and is presented by MHC2. Okay, I'm sorry, that's a bad example. Let's do that. Let's do over on that one. Let's say something funky goes on inside the cell, and you make a very bad version of some histone or something like that. That gets chopped up, put on the outside of the cell by a class 1 MHC protein. The cytotoxic T cell says, that does not look like normal histone to me, so I'm going to kill that cell. Okay, something's wrong with that cell, I'm going to kill that cell. Virus shows up, it would be processed by a professional antigen presenting cell. That piece of the virus would show up on the outside of a class 2 MHC protein, and then hopefully interact with a T helper cell, stimulate a B cell to make antibodies. Now we have antibodies against that virus. Okay. And these MHC genes are rearranged to produce the millions of different kinds of MHC that a given person would have, and that rearrangement of genes happens just like we saw in antibody production. Okay, so sorry for the screw up there. Class 1, stuff that is inside the cell is presented on the outside of the cell. Class 2, only stuff that came from outside the cell. Okay, so how do you get stuff presented on the outside of the cell? Okay, so all cells that have a nucleus, so red blood cells in humans, that when they're mature they don't have a nucleus, so they wouldn't be doing this, but all cells that have a nucleus can present antigen on their outside, but these cells, these supposedly professional antigen presenting cells, are really, really good at it. So professional antigen presenting cells could be B cells. Okay, B cells are a type of professional APC. Macrophage or a dendritic cell, those star-shaped cells that we talked about earlier when we talked about lymph, those are professional APCs, in that they are taking stuff from outside the cell and loading it up on a class 2 MHC protein, and then having that presented on the outside of the cell. Okay, so maybe it swallowed up a bacterium, maybe it took in something from the extracellular fluid just by a constitutive pathway, one that is happening all the time, called pinocytosis, so it's not receptor mediated. Maybe there is a receptor mediated endocytosis that brought in something like SARS-CoV-2 bound to an ACE receptor that's getting brought into the cell by an ACE receptor. Then it's going to get taken apart when it fuses with a lysosome, and you get these small pieces of proteins that are made. Meanwhile, MHC molecule is getting assembled over here, and it gets fully assembled and then loaded on to the antigen that comes from the proteolytic lysosome. And then this MHC molecule with the antigen, which is a piece of one of these three things, that gets then transported back to the cell surface and is presented on the outside of the cell. So a professional antigen presenting cell, which could be a B cell, it could be a a macrophage, it could be a dendritic cell. So way back here, when we said this dendritic cell, this star shape, or Patrick, the starfish shape cell, is presenting this T cell with antigen from this bacteria. That is, this dendritic cell is a professional antigen presenting cell, and it's presenting it to this T cell on an MHC2 molecule receptor. All right, here we are. Okay, along with these are these MHC molecules are toll-like receptors. And these are very good at recognizing all kinds of different antigens, and specifically antigens related to pathogens. So if there's double-stranded RNA out there, if there's parts of a bacterial cell wall out there, there is no, in eukaryotes that I know of anyway, definitely not in humans, there's no normal function for double-stranded RNA. If it sees double-stranded RNA, it says, okay, that did not come from this organism, and it'll bind to that. If it sees parts of bacterial cell walls that say that didn't come from this organism, it'll bind to that. And it can also act to stimulate these same pathways that MHC2 is going to stimulate. All right. Let's say now we're talking about MHC class 1. Okay, so this is stuff that is made in the cell and is going to be presented on the outside of the cell. And this is any old cell, not a professional APC. This is just any old cell. So your ribosome is chugging along and it makes a mistake. There's a fairly high rate of mistakes while making proteins. And what happens to that protein if it's not functional? Well, it gets tagged with ubiquitin. Our buddy, the proteasome, tears up that protein into small peptides. Before we said that was the end of the story. That's not necessarily the end of the story. Those small peptides get incorporated into the ER, where you are assembling an MHC1 molecule. That small peptide gets loaded onto the binding site of the MHC molecule, and then that MHC molecule ends up being in the constitutive pathway going through the Golgi, and ends up on the cell surface. And ends up on the cell surface with this antigen presented on the outside. Now this came from the cell, okay? And is part of what a cell is normally doing, normally making stuff. And it made a mistake, and it's going to present that on the outside. This may be from a good part of the protein or a bad part of the protein, but it's part of the normal functioning of the cell. Now that's important for something that we will talk about later, and that is how T cells learn to recognize stuff that came from the body versus stuff that came from outside the body. Okay, so this is MHC1. This came from inside the cell, so it's MHC1. It's being presented on the outside. This is MHC2. This came from outside of the cell, and it's being loaded up on MHC2, and this is only in professional antigen-presenting cells. This is any cell, MHC1. Okay. So, T cells have a maturation cycle that they go through, and they're in the thymus. This is high in the chest, this organ where T cells are maturing. They go through 2 rounds of selection, and they've got a tough job. So they have to recognize stuff that did not come from the body, okay, came from outside. And they have to also not recognize stuff that did come from the body. Because what a T cell is going to do eventually is turn on the war machine that's going to kill everything if they recognize something that didn't come from the body. If it turns on that war machine on the body itself, it gets faked out into thinking that this is an invader, but it's not. That's bad. Okay, that means that you start attacking your own body. And that happens in some autoimmune diseases like rheumatoid arthritis, where you get all the hallmarks of an infection: swelling, pain, destruction of tissue, but it's not because of an infection. It's because of T cells that are not recognizing the self. So how is it supposed to work? Okay, you make a whole bunch of MHC molecules. Okay, some of those are going to work, some of those are not. In the first round of selection, called positive selection, only cells that bind to some MHC molecule survive. So T cell receptor genes arranged randomly. They're not always going to bind to an MHC molecule made by that individual. Sometimes it's going to make something that's just not going to work. They don't. If they don't bind to any kind of MHC at all, they just don't recognize MHC to bind, then they won't receive a growth signal, and they go through apoptosis. Sort of like when we talked about apoptosis. If you don't get the growth signal at the cell surface, then apoptosis gets turned on. And that's what happens to these T cell receptors who make an MHC binding protein, make a T cell receptor that's going to bind to the MHC. If it doesn't bind to MHC, then it's no good, and they go away. So you go from this many to this many after positive selection. But then they also go through negative selection. So if this T cell receptor binds to MHC too tightly, okay, and this might be an indicator that the MHC is going to recognize self too often, or recognize self and then start calling in the cavalry, start the killing machine, kill this cell, but it's a self recognition, and you don't want that to happen. So if it has too much of a reaction to MHC molecules, MHC1 molecules are presenting self sort of peptides, then those cells are also killed. So it's only cells that have some kind of reaction to MHC, but not too much of a reaction to MHC. And then if you have some kind of reaction, but not too much of a reaction, then you'll survive, and then you'll continue to be developing as a T cell. But if you don't react at all to MHC, or if you react too much to MHC, then you're dead. You will not continue to mature. Okay. Here's one for H5P. All right. So you've got the antigen presenting cell, and you've got a T cell receptor. The antigen presenting cell has got a MHC molecule on it. The T cell also has a T cell receptor that looks just like an MHC molecule, and those two interact. So you heard me get confused before calling this an MHC molecule. It's not. It's a T cell receptor, but it looks just like MHC, and sequence-wise is similar to MHC, and it interacts. Okay? So if you have a situation where you have an antigen-presenting cell, okay, and it's presenting an antigen that is not a self-antigen, so this is MHC to class 2, okay, and it's presenting a peptide from a virus or a bacterium or something from a bacterium, okay? And this is not a self-antigen in this case. So this guy has survived positive and negative selection. It can recognize, this receptor can recognize MHC versus not recognize it at all. If it couldn't recognize it at all, it couldn't do this job. It also doesn't recognize self too tightly, but this is a non-self situation, okay, where the T cells are going to do something bad, okay? Well, bad in terms of the thing that made that, okay. So 2 signals are required to get this T cell activated. The first signal is this right here. The MHC cell with its non-self antigen is presented to the T cell receptor. Okay, that's signal number 1. Signal number 2 is a molecule made by the T cell receptor called CD28. CD28 interacts with a protein on the antigen presenting cell called CD80, and that's the second signal. So the first signal is MHC binds to the MHC receptor on the T cell. The second signal is CD80 on the antigen presenting cell reacts with CD86 CD80 or 86, depending on the antigen presenting cell, reacts with CD28 on the T cell. Okay, that's the second signal. So then a molecule inside the T cell called CTLA4 gets activated, but it only gets activated after these 2 signals, signal 1, signal 2. You get both of those signals, and then CTLA4 starts to get produced. CTLA4 gets made, and it will actually outcompete CD28 for CD80. Okay, and when it outcompetes this interaction here, right here, it's outcompeting. The red guy binds tighter than the blue guy does. That will shut off the signal in the T cell. So the T cell is activated, but only transiently until it makes enough CTLA4 that will outcompete CD80 and CD28 interaction. CTLA4 will outcompete binding for CD80, and then they'll shut off activation. So this is sort of similar to when we saw the G protein activated, but it's only activated for a short amount of time until the GTP is hydrolyzed and switched out for GDP, and then it's shut off right away. Okay. So you got an activated T cell. What is that going to do for you? It can kill a target cell if it's a cytotoxic T cell. Okay, so it gets activated by binding to a target cell. It can kill that cell if it's a cytotoxic T cell. If it's a T helper cell, okay, then it can interact with a B cell, and it'll turn that B cell into a plasma cell that will secrete antibodies. Okay, and it'll also cause the B cell and the T cell to proliferate, so you get more of this happening. Okay, they also release lymphocyte-specific cytokines called interleukins. Okay, and these white blood cell specific cytokines are proliferating immune cells, causing them to go through mitosis, and you get more of them, and maintaining those immune cells. Okay, so here is an interaction between a target cell and a T cell. This is a cytotoxic T cell. You have a class 1 MHC that is presenting something. This is to a T cell receptor. That T cell receptor says that does not look like self to me. This looks like a bad peptide. So even though it came from inside the cell, it looks like a bad dude. I'm going to make the decision to kill those. These proteases, granzymes, and perforins that generate big holes in membranes, those are released. You get a big hole forming in the target cell. The granzymes go through that big hole, and then they start caspase activation and programmed cell death. Okay, we talked about apoptosis getting started by a caspase cascade. Okay, so this is an example of a cytotoxic T cell. If it was interacting with a B cell, it would be a T helper cell, and it would stimulate that B cell to become a plasma cell. Where do we see that? Right here. Okay, and make lots of antibodies. So this T cell interacting with that B cell through their MHC and T cell receptor, that would stimulate this B cell to make lots and lots of antibodies. Okay. So, let's say you have a dendritic cell, which is part of your innate immune system. It's a professional antigen presenting cell. It acquires an antigen and it moves to the lymph node. Okay, here's MHC class 2. So this came from outside the cell, and they're not showing you the antigen, but they show it to you here. Okay, and it moves to the lymph node. This professional antigen presenting cell now gets presented to a T helper cell. Okay, that T helper cell gets activated. That activated T helper cell now interacts with a B cell that has a MHC receptor that's compatible with this T helper cell. That B cell activates a system called AID and turns into an antibody making machine, turns into a plasma cell. Okay. It could also be that this is a soluble antigen. That soluble antigen interacts with a B cell receptor, which looks just like an antibody, just like T cell receptors look like, just like MHC. Okay, and it binds that antigen, gets internalized. B cells can be professional antigen presenting cells. Presents that on its MHC, gets activated by a T helper cell, again becomes a plasma cell. Okay. In looking at what happens during a viral infection, like somebody who's getting COVID-19, after initial exposure, you get interferons being made. Natural killer cells are being stimulated. In the first days or so, you have the innate immune system involved. So natural killer cells would be part of the innate immune system. Interferons would be part of the innate immune system. The virus is entering cells. It starts to make more copies of itself. Then the immune system hopefully catches up. Okay, maybe about a week after the virus has entered the body, you get antigens presented from that virus now presented to on B cells or dendritic cells, and those B cells start to get stimulated and turned into plasma cells, and they start pumping out antibodies, and the amount of antibody goes up. And then those antibodies either are neutralizing antibodies, they will bind to some part of the life cycle of the virus and prevent the virus from going through that life cycle, like maybe binding to the spike protein on the coronavirus, and or they are identifying cells that are attacked with virus so that macrophages can identify those cells and kill them. If you get re-exposed, we haven't talked about this, but there are memory cells that are made in this activation of T cells. They can be rapidly go through mitosis, and you get rapidly pump out more antibodies more rapidly than the first time you're exposed, and so the virus titer comes and goes quicker when you're re-exposed. Okay, so that's a lot, and but hopefully you can see how it dovetails with some of the stuff we've been talking about since we've been online here, some of the coronavirus stuff and some of the other pathways that we've been talking about. I want to give you an update on some of the coronavirus science news. There was a remdesivir result released in New England Journal of Medicine, where there is a part of clinical trials called compassionate use, where if someone is near death and the drug might help them, they are allowed to use it. It's a very small number of people in which they allow this. They have results back from only 53 patients, and about 68% of them improved as a result of this remdesivir, which is a drug that had been tried in Ebola and was not effective, but it was effective against other types of SARS viruses. They also, later on in this paper, you can take a look at it if you want, there are some very serious side effects of these drugs. So they're not fun to take. It's not like taking an aspirin or something like that. I have talked about antibody testing. I've talked about it on the radio. And one thing I didn't really understand until this week is that probability makes a big difference in how these things, how to interpret the results from these antibody tests. So I'm going to explain this, try to explain it quickly because it's already a long podcast. But if you hear that a test is, say, 95% reliable or something like that, that sounds fantastic, right? Anybody would be happy with a 95 on a test or something like that. 95% is really good. And it is really good. But it all depends on what percent of the population has the virus. So if the virus is relatively rare in the population, then that means the probability of finding someone who has been exposed to the virus in a random sample is low. So let's say at 1% of Ohio citizens have been exposed to this virus. So if I tested 100 people, by chance only one of those people would have the virus, okay? And let's say that the test is 95% reliable. That means if I tested 100 people, that 95 times out of 100, I would get a correct test result. But 5 times out of 100, I would not get a correct test result. So it may say that, yes, you have been exposed, when in truly you haven't been, for 5 people out of those 100. Or it may say, no, you haven't been exposed, when truly you have been, for 5 people out of that 100. So when the prevalence is rare, and the false positive rate is as low as, say, 5%, okay? And again, that would be the case if you had an antibody test that was 95% reliable. Then it's possible that the result from that antibody test that you get could be wrong like 85% of the time. just by how the probability works. And so I've even said this on the radio, that it was sort of like you get your antibody test, and if it says you've got antibodies, then that's a get out of jail free, and that was wrong. It is not a get out of jail free. First of all, we don't know, it just tests for antibodies. So even if the test is telling you that, yep, you have antibodies against COVID-19, and even if though that's true, okay, and because of the probability thing, we don't know that's true, but let's just say that it is true. We don't know necessarily that those are neutralizing antibodies. It just says antibodies. So it could be that it making antibodies against the COVID-19, but those antibodies are not enough to protect you. And so if someone has a positive result from an antibody test, they should still be as careful as they would be even if they didn't have that test. So if they are a healthcare worker, they should still take all the precautions, wear all the PPE that they can get, if they can get it, even though they have been exposed. And so the antibody testing, we should be doing it, and we should be doing it at a much higher rate. But it's not, we need to know more in terms of what is the background exposure to know how reliable those tests are going to be. Today's podcast, you saw that interleukin, the lymphocyte-specific cytokines get released. And as part of the complement system, you see all kinds of things getting released. And one of the problems going from a mild infection to a very severe reaction in COVID-19 is so-called the cytokine storm, what we talked about before, in that the immune system is called to an area and they release all the things that the immune system cells release that are designed to kill cells. And if that happens to too great an extent, it kills the organism, the patient, instead of just killing off those cells. So you could theoretically block the immune system and block the cytokine storm. So if you're having a too severe immune reaction, you can tamp down the immune reaction. And this is something that is done in autoimmune diseases where people take steroids, that sort of thing. I won't take you to that link, but there's a link right here where they're using artificial intelligence to identify potential blockers of this cytokine storm. There are physicians who are very wary of this, though, because often what's happening in COVID-19 patients is that they have a secondary infection. So pneumonia is a bad thing when your lungs fill up with fluid, and there can be opportunistic bacteria that causes secondary infection. And if you are knocking down your immune system to block the cytokine storm, then you don't have any immune system left to fight the secondary infection. And so even though this is being pursued as a possible treatment, even has been used as a treatment in some cases, there are physicians worried about it for that reason. Okay. So, are we getting near the peak? So the governor is talking about opening up the state. Are we getting near the peak? So I've been looking at these every day, and they're published by the state. And if you just look at the number of cases, it just may be tapering off. There's only a couple days on there, and we'll get more next week. But we could be near that peak, or it could be just a shoulder like this, and then we'll go back up. We will see. There's also one of the things that is being tried is custom recombinant antibody design. So there's this little
This is the story about how we find antibody medicines. First, we take some genes that are the molecular recipe for antibodies and combine them on a strand of DNA. We package the DNA in a protein coat made from a virus which only infects bacteria. The antibody genes make an antibody hat which is attached to the top of the virus coat. The virus is called a phage, and the combination is called a phage antibody. The phage antibody hat is unique and gives it the ability to bind to a disease target molecule. Change the antibody genes, and you change the type of hat and what it can bind to. We make many of these antibody phage, and our pool has over 100 billion different kinds. Together, all these antibody phage are called a library, and this is our source for finding new antibody medicines. To target a particular disease molecule, we therefore need to find the right phage antibody from our library. For example, if we want to find an antibody to this orange one, we can use it as bait, like a fly on a fishing rod. Only the antibody phage that has a hat which can fit the orange shape will bind and stick to the target molecule. We can fish in our pool of antibody phage with our disease target and pull out each antibody phage that bites on our bait. When we find one that binds, it brings the antibody genes with it inside the coat. Because the genes are made of DNA inside a virus, we can use bacteria as hosts to make more of the virus and replicate it. We then take the genes from the antibody phage and, along with other genes, combine them to make instructions for a whole antibody medicine. Antibodies are quite complicated, so we need to ask cells to act as factories to make them for us. We can then collect the antibody medicines from the cell and use them to treat disease.
Okay, so this approach is called phage display, and essentially what you're doing is replicating the maturation of B cells in a test tube, and then you're making a library of potential antibodies, and then you can test that against, say, COVID-19. It is being used as an approach for COVID-19 antibodies. It can be faster than making antibodies in, say, a rabbit, and they're working on it. This company is working on it right now. And then finally, I wanted to mention there are some testing results from, you heard about this aircraft carrier in Guam, where there was some COVID-19 cases. Very hard to social distance on an aircraft carrier. The commander got removed from command, but they've now tested almost all of the soldiers on that carrier, about 4,800 of them. There are over 600 people who are positive for COVID-19. And what is really surprising from these data is that 60% of those positives are asymptomatic. So the highest figure I had heard before these data were that as many as 25% of people walking around are asymptomatic. 60% is really high. Now, in that population, there's going to be more people who are young than in the whole population. Yet that is a really high number, and it's a pretty big sample size. So people are really rethinking how we need to think about this in terms of building our models for how to open up, and how we really need much more testing of our population to get an idea of who's infected and who's non-infected. And probably at least 10 times more testing than has been done so far to get a good idea of that. All right. Thanks very much.